Ladies and gentlemen, my name is Stalis Norma Ethica from Indonesia. I work as a full-time lecturer in the postgraduate program Magister of Clinical Laboratory Science, Universitas Muhammadiyah, Semarang. I'm presenting this keynote speech today for the 3rd International Conference on Research in Life Science and Healthcare, held in London, April 29, 2022 by HBSRA. This keynote presentation is sourced from my current research and it's entitled Brown Algae as a resource of novel alginate-lyase producing bacteria to combat biofilm-related infection. Here I'm showing you the introduction. Current status of global infection resistance in the world as reported by Srinivasan et al. 2021. According to this report, in 2019, more than 1.2 million people and potentially millions more died as a direct result of antibiotic-resistant bacterial infections. According to the most comprehensive estimate to date of the global impact of antimicrobial resistance, or AMR. Also, the analysis of 204 countries and territories published in the Lancet reveals that AMR is now a leading cause of death worldwide, higher than HIV, AIDS, or malaria. This report shows that many hundreds of thousands of deaths now occur due to common previously treatable infections such as lower respiratory and bloodstream infections because the bacteria causing them have become resistant to treatment. As you can see here, the red area shows the highest occurrence of this infection resistance in the world. Interestingly, biofilm is among the prominent factors in causing pathogenic bacterial resistance. There are two main groups of biofilm-related infections. So, the first is device-related, such as ventricular derivation, contact lenses, endotracheal tube, central vascular catheter, pacemaker catheter, etc. And the second is tissue-related, such as chronic sinusitis, ear infection, dental plaque, endocarditis, etc. This figure from Shrestha 2021 lists the type of biofilm infections belong to the both groups. Due to their high tolerance towards antibiotics, these device-related and chronic tissue-related biofilm infections are difficult to treat and expose the patient to the risk of recurrence. So, what is bacterial biofilm? So, it's a clusters of bacteria attached to a surface and or to each other and embedded in a self-produced matrix. So the blue part here is the self-produced matrix, and it contains high level of alginate polymer. Biofilms may involve in up to 80% of global infections. Biofilms here protect bacterial cells from antibiotics and disinfectants causing the resistance. So I say again here, main component of bacterial biofilms is alginate. The blue part of the left figure shows the bacterial self-produced matrix, which actually contains also water and extracellular polymeric substance, or EPS, extracellular DNA, lipids, and extracellular vesicles. An example of bacterial biofilms in chronic wounds is displayed here in the right part figure, and biofilm can also be found in catheter surface, dental plaques, as I mentioned before. This according to Wounds International. Bacteria in chronic wounds, for example, are protected by biofilm, which induce and exacerbates the inflammatory phase of wound healing. Here I'm showing you the biofilm development steps. So the progress is basically in a cycle. The microbial biofilm cycle could be classified into 4 phases, such as initial attachment or adhesion, and then adherence, maturation, and dispersal. The biofilm inhibitory and dispersal strategies are summarized in the below figure. As seen in this picture, so the A step, the initial attachment, can be disrupted by interfering with the interaction between the surface and the microorganism, either by surface remodeling or by physical removal of the biofilms in this part, the A, yeah. And then B, adhesion can be inhibited here by targeting biofilm EPS and cellular division. In step C, disruption of biofilms in proliferating and maturing phase may be accomplished either by physical removal or damaging the EPS matrix, primarily by affecting the formation of pathogenic microenvironments such as hypoxia or low pH, and quorum sensing along with the eradication of persister cells. In D part, biofilm dispersal could be achieved by remodeling the EPS matrix or accelerating the dispersal mechanisms. Here in the picture, different colors of the cells represent different bacteria within the biofilm. Circular cells represent cocci and rod-shaped cells represent bacilli. Here is the diagrammatic representation of the alternative approach against antibiotic-resistant biofilms communities, as summarized by Sharma in 2019, is seen in this slide. So as we can see here, among important approaches are photodynamics therapy, and then CRI-SPR SAS9 technique. The CRI-SPR stands for clustered regularly interspaced short palindromic repeats, and the CRI-SPR associated protein 9. And then nanoparticles, anti-quorum sensing signaling molecules, and then natural compounds, among which is alginate-lyase enzyme that we are talking about in my research, which works by degrading the biofilm matrix. And actually, I want to explain here why we are interested in the marine environment, because the environment offers new compounds, including antibiofilm agents. In this slide, I'm showing you my group's research interest, marine brown algae. It is a rich source of alginate. It means that the content of alginate in brown algae is actually higher compared to that of red or green algae. And bacteria degrading alginate in brown algae is called Alginolitic Bacteria. These bacteria degrade alginate by secreting alginate-lyase enzyme. This is the alginate-lyase enzyme as modeled by computer. Why interested in marine environment again? Because marine environment is a harsh one, with high salinity, high sun ray exposure, and temperature extremes. And this makes marine enzymes adapt to such harsh environment, making them have unique yet beneficial characteristics such as resistance to temperature and pH changes. And these characteristics are preferred by the industry. So how is the mechanism of biofilm degradation by alginate-lyase enzyme from Alginolitic Bacteria of brown algae is displayed here. Biofilms are known to be resistant to antibiotics, even to disinfectants such as chlorine. Why? It is because pathogenic bacteria in biofilm are protected from any antibiotics and disinfectants. And because of such a nature, the use of antibiotics alone is not effective in treating biofilm-related infections. One of important strategies to kill pathogenic bacteria is by degrading this biofilm and then exposing them, the bacterial cells, to the killing agents. This will make the use of antibiotics and disinfectant as the killing agents more effective, so can reduce their dosage for the same amount of targeted pathogenic bacterial cells. And the summary of my current team research project is actually screening or isolating alginolitic bacteria from marine environment, and then purifying and characterizing the obtained bacterial alginate-lyase. After that, we want to overexpress bacterial alginate-lyase coding gene in order to scale up the production of enzyme. Finally, to obtain new antibiofilm agent from hopefully novel bacterial species. Well, actually, we up to date, we have collected 3 novel bacteria producing alginate-lyase, and we have done the whole genome sequencing of the 3 bacterial isolates, but we haven't published them yet. And again, this agent will accompany the commonly used antibiotics in the effort to eradicate infectious or pathogenic bacteria. By doing this, the amount of antibiotics could be better controlled, not overdosage. So that's all for me. Thank you for all of your attention. Have a good day.